Okay, so what I'm going to try to do for you guys is for anybody who couldn't get the simulation to work, I'm going to walk you through watching mine. And basically, I'm going to just tell you guys when you should stop. So, this first one, I'm going to just have you guys pause your video and read this first slide. And then if you follow along in the doc too, there's certain things that you should put in your notes or put in a doc to keep. And this is one of them, kind of keeping in mind, this overall process. DNA transcription makes mRNA, and then from the sequence in mRNA, you make a protein. So make sure to have that down in your notes somewhere. The second part, you don't need to spend a ton of time on. So basically, it's just a model of DNA. And our question here is, how does the sequence of nucleotides allow us to make a protein? And so you can look at this in different ways. You can look at sticks. So these are the individual, you can find the sugar phosphate bases in there. You can look at them, kind of move these around. But anyway, you don't really need to do that. You don't need to take any of these pictures. So I'm going to move on to the second one. And I'm going to let you guys pause the video and just look at this for a second. And so the key here is kind of getting an idea of what transcription is. And actually, I think I clicked on this before. I don't actually think that that's transcription. There's one of them that didn't work. But so down here, what you're going to do is model transcription. And so keep in mind this is in the nucleus, and this is DNA. And to make a copy of it, or to copy the message, you have to first separate the strands. And then what you're trying to figure out is what would be the complementary bases that you would put in here. But then remember that these are RNA. And so if you try to go through here, so you'll say, what's the complementary base of thymine, adenine, cytosine. So what you can do for just a second is, try to maybe write in your notebook or write on the whiteboard, what do you think this sequence is going to be? But then, so if you want to do that, just pause for a second. But then what I'm going to do is, so you can actually put in, so that's the complementary base. And then the difference, you can see here, DNA has CAGT, and RNA doesn't have thymine, it has uracil. But uracil is complementary to adenine. So when you're making RNA, it's going to be complementary, but the only difference is there's no T, there's going to be U in its place. So hopefully you're getting the hang of how to do this now. Okay, so now you have your RNA. And then the point here is that RNA strand is actually going to detach and will be the message that you use for translation. So take a second and read these two. So the bottom strand is transcribed. Which DNA strand is most similar to the RNA strand? And if you look here, so this AUG, UUG, it's pretty close to this sequence. AUC, the only difference is the T's and the U's. And then it says a group of 3 nucleotides codes for 1 amino acid. Notice the black tick marks right here above the DNA strand showing the triplets, which are called codons. So how many amino acids are coded for by the strand in the model above if each 3 of these codes for 1 amino acid? And so hopefully if you count those up, so you have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. All right, so this is one of the things I think you're going to put in your, what are the rules about complementary base pairing? And then for this strand, what would be the complementary bases? Okay, so we're going to go on to section 3. All right, so now this is all about translation. And so this is taking the RNA and basically translating it. So taking a molecule that's a nucleic acid and then translating what does that code mean for an order of amino acids that will make a protein. So I'll let you read this for a second. You can pause. And then so now what we're going to do when you're ready is model transcription and translation. And so here, what you're going to do is first transcription. So DNA breaks apart, fills in the RNA, the complementary base code, and then, so you're going to take that RNA and translate it. So every 3 nucleotides, you're going to put in 1 of the correct amino acids. So when we do translate, so each 3, 1 amino acid gets put in. And then what will happen at the end is this string of amino acids, they start to interact with one another, with the water outside, and then this folds up into a 3-dimensional shape. So what you can do is, they describe this down here. Anyway, so, and actually I think the video that I gave you guys for the second class will actually help with this too. But you want to keep in mind what happens at each step. And so actually let's do this. So we can move that. So this is a protein. It was a string of amino acids and then it folded up. So what you can do is you can do this again. So you can transcribe, and if you wanted to, you could do this step by step, but that's what we just did before. And then when you translate step by step, so this green thing is the ribosome, and that's sort of what organizes everything. And there's one piece that you actually don't see in here, and we're going to go to that in a second. But at each step, so every codon, the right amino acid will be put in, and then the next codon, the right amino acid. And we'll find out how to actually translate those a little bit later. So each codon, the right amino acid gets put in. And then this even happens in real life. This happens as this is being made, but when it's finished, then these amino acids start to interact and they start to fold up into a protein. And so one of the questions I think that I asked was, compare the sequences and the shapes of the different proteins produced from the same sequence of RNA. And so just think about, so we can stop this, we could even do this again. Oops. So I'm going to transcribe it again and translate and think about how does the sequence of the amino acids and the shape of the protein compare when you make a protein from this exact same sequence. So hopefully you have a sense of how this works, and then you're going to look at some more details about how this process actually works too. You can kind of see how those fold up. You might want to talk about the obvious. So this glutamine and serine, they're always on the end. Maybe look for, are there any other similarities with how these fold up? So what's missing here is how do you actually put in the right amino acid? And so this is your mRNA, this is your code, and there's this molecule called a tRNA. It stands for transfer RNA, but I think of it also as the translator. So you have this part here, it's called an anticodon, but it's 3, so this is RNA, so these are 3 nucleotides that complementary base pair to the sequence in the mRNA, and on the other side of the tRNA is the right amino acid for that code. So these are like little translator molecules that read 3 bases and say what amino acid should go in that spot, and then they connect that amino acid to this chain of amino acids, and what you get is the right sequence of amino acids for the right sequence of nucleotides in the mRNA. All right, so let's see what else is there. Okay, so I think we're on to page 4 here. Okay, so how you actually, so it says now it's your turn to perform the work and translate the DNA sequence. And so the way that we do this, so these are representative of codons, And so every time you have, so this codon, uracil, 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 the same amino acid gets put there. So every time you have cytosine, uracil, uracil, in the mRNA, there's a leucine that's put there. And so keep in mind that these codons are always the mRNA. And the way that you can read this chart is, so let's say your codon was AUG, which happens to be the first one usually. So your first base is A, so you're going to be in here, in this row. Your second base is U, so you're in this box. And then you look for AUG, and so that's methionine. So let's see, if you go down here, so you're going to say, all right, this is the mRNA. So that's so you can tell if it has T, it's got to be DNA. So we're looking at the mRNA. And so you're going to do this, AUG, so A, U, G, and then you just click on that one. The next codon, UUG, U, U, G. All right, so actually what you should do is pause the video and try to translate the other codons. So every group of 3, try to figure out what amino acid goes there. So you can pause right now and try to do that. And then once you're unpausing, you can kind of follow through and see if you got these right. So we've got ACU, so that's threonine, then CUA, and then ACU, and then CGC, CGC, and then AUC, and then CGC, ACU, G, UG, and then UAG. So if you go UAG, you're going to see this stop, and that actually just means there's not an amino acid that goes there. It just sends, well, a molecular signal, but it basically stops the synthesis of the protein. So then it breaks off, and then it starts to fold up. So so you can look at those questions. Yeah, basically, can you translate a protein? And we'll do some more practice with this later. So let's go to 5, and actually I might stop the video soon. But what you can look at is a mutation, so a change in the DNA sequence. So if you'll read through this and you're looking for what is a mutation and what causes a mutation. And then ultimately, the next couple pages are describe how a mutation can change the shape and a function of the protein. So you can stop and read this. But so some of these are making predictions about mutations. So number 6 is that you can make some mutations in the DNA and then see what happens to the sequence. So I think this is going to make more time. I'm going to stop the video now, but if you want to do this, so the most important piece has already been in the video, right? How do you take a sequence of DNA and make a sequence of amino acids and a protein? But so keep in mind that the point of this is that if you change the DNA sequence, then you could change. You don't always, a change doesn't always result. But if you change the amino acid that goes in there, you are going to change the sequence, which could have changed the shape and the function of the protein. All right, hope this helped if you couldn't get your simulation to work.